And let me start off with a clinical example. This is the cardiac MRI from a young patient, male patient in his 20s, who obviously suffers of severe dilated cardiomyopathy. Likely familiar. We were not able yet to identify the genetic cause, but I'd like to take that as an opportunity to also discuss, as you've heard already today, that the etiology and also the progression of the disease of dilated cardiomyopathy in contrast to hypertrophic cardiomyopathy is much more diverse. So as you have heard, the sarcomere, of course, plays also a role in dilated cardiomyopathy, however less than in hypertrophic. Mitochondrial and energy mechanisms play a role. The sarcolemma plays a role, aberrant calcium cycling, and even nuclear mechanisms play an important role in the pathogenesis of dilated cardiomyopathy. And yet I'd like to convince you today that even more cellular mechanisms may also participate in the progression of dilated cardiomyopathy. So why talk about protein degradation pathways in cardiomyopathy? Just as a reminder, there are several pathways the cell has to degrade in a controlled fashion protein, for example, misfolded proteins. One major mechanism is the ubiquitin proteasome system, as all of you know. I'll get back to that in a second. And another important mechanism is the autophagy and lysosomal pathway. Again, both of these pathways have the purpose of degrading protein or even entire organelles. So what's the relevance for cardiomyopathy and even human heart failure? Well, there's one paper from 2008 that could nicely show that DCM samples, heart samples from human patients, have a remarkable increase of ubiquitination, just in general of all proteins, and you can quantify several key components of the ubiquitin system, E1A and E1B. Again, there is a significant increase in the activity, the overall activity of the UPS system. And as Lucy Carrier has shown, and she's here in the audience, this is also relevant for several of the sarcomeric proteins we've heard about today, and I'll get back to that again in a second. So there are the first components already known, how specifically sarcomeric proteins are degraded in cardiomyocytes. So apparently the UPS plays an important role also in cardiomyocytes, as in any other cell. How about autophagy? Again, similar data, so also human samples, myocardial samples have been taken and analyzed for key components of the autophagy pathway. For example, beclin or LC3-2, I get back to that also in a second. And you can see that hearts that are unloaded and upon LVAD implantation, they have a drastic differential regulation of these key components of autophagy. So in a loaded heart, you have a rather high level of autophagy, and that is down-regulated again upon unloading. And Joe Hill's group, who's done a lot of work on autophagy, has summarized that in a review article where several models, even human patients and also animal models, could be shown that cardiomyopathy is typically associated with an increase in autophagic activity, and unloading of the heart, again, decreases this. So what is our approach to this story? So you've already heard a lot about genetic testing, about next-generation sequencing. We kind of took a reverse approach. So we figured that most of the genes that are known to cause or play a major role in the pathogenesis of cardiomyopathy are expressed in a more or less heart-enriched or even heart-specific fashion. So we set out to find genes, novel genes that are typically just specifically expressed in the heart. And we did that in the database just 10 years ago. We identified about 40 of these genes. This is just an example. You see that all these so-called expressed sequence tags, so short tags and expressed cDNAs, are all from the heart in this specific gene. And then, of course, you have to validate this assumption that you have a novel cardiac-specific genes. You have to do northern blots, and these are just a few examples of novel genes that are all more or less specifically expressed in the heart. And then we designed a cloning platform where we cloned these novel genes that at this point we didn't know much about into several vectors, adenoviral vectors to be able to overexpress them in cardiomyocytes, microRNA vectors to be able to knock down the gene, even prokaryotic vectors to generate antibodies, and yeast-two hybrid vectors to identify protein interaction partners. I'd like to show you two examples of novel or uncharacterized genes. One of them is called MYFbx or FBXL22, which in our view plays a major role in ubiquitin proteasome signaling in the heart. And secondly, another gene called FICO1, which plays a role in autophagy. So first, MYFbx. So I've just shown to you that the ubiquitin system plays a role in any cell and also in cardiomyocytes. It's a very regulated, stepwise approach where several enzymes, E1 to E3, lead to the ubiquitination of target proteins and then subsequently to the degradation of these pathways. And the specificity of this pathway is typically achieved here at this step, the E3 ligase. So there is the substrate specificity. Of course, you don't want to degrade just any protein. So MYFbx, this novel F-box protein that we identified by bioinformatic means, is indeed expressed in the heart and almost exclusively in the heart, as you can see in these northern blots, both from human and mouse tissues. This is an antibody, so in western blot. It's a rather small protein, and it has a very conserved N-terminus, which is the so-called F-box, a structure that is known to be part of an E3 ligase, also in other proteins. And just to look at the other species, you see that even down to zebrafish, and I'll get back to that in a second, the N-terminus is highly conserved of this novel cardiac-specific protein. So we first asked, well, what could be the interaction partners for this novel protein? And we performed a yeast-two hybrid screen, which is able to identify in a library additional and novel interaction partners. And we were pleased to find that the first clone we picked was a protein called SCIP1. Many of you are not familiar with that, perhaps, but it's a key protein in the ubiquitination reaction. So SCIP1 is the protein that binds to the F-box of E3 ligases, and this entire complex, and I don't want to get into the details, subsequently leads to a substrate-specific ubiquitination. And keep in mind, the F-box protein, in this case, we believe MYFbx, is responsible for the specific binding of substrate in a given cell type. And, of course, we were expecting from this yeast screen that we might perhaps identify interaction partners that could be substrates for this novel F-box proteins. And indeed, we found two sarcomeric proteins, actinin 2 and filament C, as direct interaction partners for MYFbx. We could confirm the binding of these putative interactions by co-immunoprecipitations, just shown for actinin and filament C. And as you would expect from these data, we could also show that FBXL22 or MYFbx is indeed localized to the sarcomeric Z-disc, as you would expect as actinin and filament are known Z-disc proteins. And just a reminder, and you've seen similar data just a minute ago, the Z-disc is believed to be one of the hotspots in the pathogenesis of human cardiomyopathy. Several proteins localized to the Z-disc have been shown to be associated with cardiomyopathy, and this has recently been summarized in this review article where all of these Z-disc proteins have been implicated in either cardiomyopathy or myopathy. And just as a reminder, and I've seen a poster today that shows similar data, this is also true for actinin. So actinin is also a disease gene, if rare, but a disease gene for dilated cardiomyopathy. So how about the function of this novel protein? Is there any relevance for this interaction? And we speculated that indeed MYFbx might be mediating a degradation of the target proteins actinin and filament. So we first did an overexpression experiment where we overexpressed both actinin and this novel F-box protein. And as you can see here, the increasing dosages of the F-box protein, MYFbx, there is a degradation of actinin 2. And the same is true for filament C. So increasing dosages of the F-box protein lead to subsequent degradation of filament C. And this is also, as you would expect, associated with an increase in ubiquitination. As you can see here, in the presence of the F-box protein, there is a slower migrating smear, which is typical for increased ubiquitination. And another proof of this concept was to inhibit the proteasome by a drug called MG132, which is a potent proteasome inhibitor. And while we saw, again, degradation, in this case of filament in the presence of MYFbx, in the presence of this inhibitor, there was no difference in the expression level, suggesting that indeed the degradation is proteasome dependent. Finally, we also did the same experiments in cardiomyocytes, so the relevant cell type. We generated adenoviruses overexpressing MYFbx, and again we saw actinin degradation in cardiomyocytes in the presence of MYFbx, and this is the quantification. And finally, we also ask, is the opposite true? So when we knock down the F-box protein, does that perhaps lead to accumulation of actinin? And that was also the case. So when we degrade FBXL22 by a microRNA, synthetic microRNA, there's an increase in the actinin concentration in cardiomyocytes. So it seems that this novel protein controls the turnover of actinin in both directions, either down or upregulation. So again, you may ask, so what's the relevance for disease here? So we ask whether perhaps in vitro and in vivo models of hypertrophy and failure in cardiomyopathy may lead to a differential regulation of this novel protein. And we were quite impressed by a dramatic downregulation of this novel protein upon hypertrophic stimulation. These are just cardiomyocytes exposed to phenylephrine. But the same is true in mice that have been banded, so a pressure overload model, again, a marked downregulation of the MYFbx protein. And since we tested hypertrophy models, we again asked, well, maybe this novel protein is able to modulate hypertrophy itself, and we tested several hundred cells by measurements, and we could show that at baseline there is no difference in MYFbx treated cardiomyocytes. As you would expect, phenylephrine treatment leads to an increase in cell size. However, simultaneous treatment with MYFbx overexpression leads to a complete abrogation of hypertrophy, suggesting that the controlled degradation of actinin and filament somehow prevents hypertrophy. And finally, we also performed an in vivo model in cooperation with Wolfgang Rottbauer in Ulm, who did a zebrafish model, now a knockdown of this novel protein. You see here a control zebrafish, which pumps nicely, and just compare the knockdown of MYFbx. You see a severe phenotype with a marked contractile deficit in these zebrafish. And if you quantify this effect, it's very reproducible, so about 90% of the treated fish have this cardiomyopathy phenotype, and you can even quantify the fractional shortening in this model. It's already at 48 hours post-fertilization. There's a marked decrease, and that's almost a complete cardiac standstill after 72 hours, so severe cardiomyopathy. And last slide about MYFbx. So what could be the mechanism? You would predict from the downregulation of MYFbx that the target proteins should accumulate, and at least for actinin, this is the case. So already at baseline, there's a marked increase in actinin concentration in these zebrafish hearts, and if you treat the animals with the proteasome inhibitor, you can even amplify this effect, so there's even more accumulation of actinin, and we'd like to speculate that the accumulation of actinin or other target protein is the cause of the cardiomyopathy in this model. And the specificity of this effect could be shown by looking at several other sarcomeric proteins which were not altered in this model. So in the second part of my talk, I'd like now to switch gears and to talk about autophagy, the other major pathway in protein degradation. Again, just a reminder, so protein or even entire proteins that are, for example, misfolded, that are bound to heat shock proteins, or that are even entire dysfunctional organelles can be subjected to so-called autophagy, forming organelles that are finally fusing with lysosomes and thereby kind of leading to a recycling of cellular components that are, for example, useful for energy production. So what is this novel or uncharacterized protein FICO1? Again, we found it by relative cardiac and skeletal muscle specificity. You see here the expression levels in heart and skeletal muscle versus all other organs. And this is a northern plot that again shows that it's almost exclusively expressed in striated muscle tissue. Again, we performed a Yeast-two hybrid screen to identify potential interaction partners, and we identified a protein called ATG8 or LC3, which is a known component of the autophagy pathway, so that's why we figured it might have a crucial role in this process. Of course, I don't want to confuse you with this detailed slide, but you get the message that it's a very complex and highly regulated event, a signaling event in the cell, the autophagy formation. And just suffice it to say that a key component of this process is mediated by LC3 or ATG8, which is important in the final steps of autophagy, so the forming of phagosomes and finally degradation of these cellular organelles. So as we found that FICO1 directly interacts with this LC3 molecule, we again analyzed this now in cardiomyocytes. We generated adenoviruses overexpressing FICO, and we're again pleased to see that FICO perfectly overlaps by immunohistochemistry with LC3 staining, so consistent with the idea that those indeed interact, and we could also do co-immunoprecipitations. But how about the function? Is that relevant for any pathophysiological process? Well, it is. There are several papers, and these are just two of them, who could show that various treatment, in this case energy starvation of cardiomyocytes, leads to a marked induction of autophagy. And this is measured by the increase of the LC3-2 isoform. So there is a shift of isoform, and this is generally accepted in the literature as the marker for autophagy induction. This is shown in several different papers. So starvation leads to increased autophagy as measured by LC3. Same here in this other paper. So starvation leads to recycling of energy. That's the idea, to keep the cell alive. So how about FICO? Well, we tested FICO now in vitro in cardiomyocytes, overexpressed this molecule, and tested again for this readout of autophagy, so the shift towards LC3-2 isoform. And you see here that already at baseline, so under good nutrition with normal glucose, there's an increase in autophagy, but this is even more dramatic and amplified in the absence of glucose. When we then treat cells with FICO, there's a really dramatic increase in autophagy. This is the quantification. If we do the opposite experiment, so knockdown FICO, you would expect that perhaps the autophagy pathway is regulated in the other direction. We first had to show that our synthetic microRNAs are indeed able to knock down FICO. This is the case, so both microRNAs completely abolish FICO expression in cardiomyocytes. And when we treated cardiomyocytes that are both treated with glucose or without glucose, we could show that without FICO, there is no autophagy going on anymore, even though the cells are deprived of glucose. So it's apparently a potent regulator of autophagy in vitro. Of course, we were also wondering, well, does this have relevance for the in vivo situation? We generated a mouse knockout model to be able to study this question. The knockout was working, so the knockout animals do not express FICO anymore. And at first we were disappointed since the baseline phenotype of these animals, perhaps unexpectedly, is quite normal. So the fractional shortening, the left ventricular diameter, heart weight body weight ratio as an indicator for hypertrophy, and even expression of hypertrophic marker genes, A and F, B and P, is completely normal in these knockout mice. Yet, when we then stressed these mice, and the first obvious stress experiment was starvation, and it's quite remarkable. You only need to starve mice for 24 hours, and you already get a severe phenotype. So the starved FICO1 deficient mice develop contractile dysfunction, again only after 24 hours of starvation, with a marked depression of fractional shortening, and this goes along with the biochemical marker that, again, the autophagy marker is repressed in the knockout model. How about a more relevant or disease-relevant model, pressure overload? Again, it has been shown before by others that autophagy is indeed increased in the heart upon pressure overload. You can see that even on the electron microscopy level. Here are these nice phagosomes, and this is a robust increase in the LC3 induction already after 24 hours and over the subsequent weeks upon banding and pressure overload. So how about the FICO knockout? So again, we also subjected our animals to pressure overload. There was a slight decrease in hypertrophy, not impressive, however significant. So FICO1 deficient mice develop less hypertrophy compared to wild-type banded animals. However, the effect on fractional shortening was more dramatic, and now we have older animals, the difference is even larger. So wild-type animals already develop a depression of contractile function upon pressure overload, and this effect is even more pronounced in FICO-deficient mice. So suggesting that, at least in this model, the absence or the inhibition of autophagy is maladaptive, it's not protective. I'd like to summarize, as we've heard in many talks today, the pathogenesis of cardiomyopathy and heart failure is of course complex and still poorly understood. We believe that the characterization of novel candidate genes may reveal insight into the mechanisms of heart failure and perhaps on the long run provide new therapeutic targets. The ubiquitin proteasome system, or UPS, and the autophagy lysosome pathway play an important role in cardiac physiology, and I hope I convinced you also in pathophysiology. And finally, these new proteins, MYFbx and FICO1, are two previously uncharacterized proteins that are involved in myocardial protein homeostasis, and the first in vivo data indicate that they may have an essential function in the adaptation to hemodynamic stress. Thank you for your attention. I'll close with the acknowledgement of my team in Kiel. Thank you very much.
Thank you. Very, very interesting. We talked about these systems just before the meeting. We had the local meeting of the European network, and with Lucie Carrier, who's obviously an expert here. But I would like to hear your opinion as well. How much you're obviously speculation, but in genetic cardiomyopathies, could a variability in these mechanisms, individual variability in this mechanism, partly explain why you have such diverse phenotypes in the same family?
As you said, I can only speculate, but I'd like to think in this direction. So we tested these genes also in our patient cohorts, and we were not able to at least convincingly find mutations in FICO, for example. We have some that are potentially pathogenic, but we don't really believe in that, and we have no familial case. And I'd like to believe what you just said, that they perhaps have a disease-modifying role, and this is likely, since they also play a role in heart failure per se, not just in genetic dilated cardiomyopathy. But we need more data to really answer that.
Question? Very nice data. I actually wanted to follow on this point. Did you have the chance to analyze the expression level of these two proteins, mRNA and proteins, in patients with hypertrophic cardiomyopathy? I mean, in septal myectomies or explanted samples from DCM patients, heart failure samples?
Actually, we haven't, and that's a very good idea. So perhaps we should talk about that afterwards. I mean, given your data that in HCM, especially myosin binding protein C mutations, these mechanisms play a role, you may speculate that these are differentially regulated, even in the septum. So we should check that. Mm-hmm.
I actually had a couple of questions, if I can. So the first F-box protein. So so you have data on the animal model, it's just a zebrafish, so it's a kind of a primitive type of model. But having a hypothetical mammalian model, would you expect more a dilated phenotype or a right ventricular cardiomyopathy phenotype, considering the proteolytic pathway? And the second question is there any link between the second protein, the FICO1, with the Danon disease and the LAMP2 protein?
Yeah, maybe I can ask the second question first. So there are, as you all know, there are some cardiomyopathies that are protein, aggregation diseases, such as Danon or also desmin cardiomyopathy. And in that case, autophagy is impaired. And you may even think that therapeutically it may be useful to increase autophagy, to get rid of these aggregates. However, in other instances, too much autophagy is also maladaptive. So it's probably a matter of dosage. But in that case, it probably plays an important role. And your first question about another model, so for MYFbx, we are currently generating a knockout mouse, but I can't really tell you. I mean, in my experience, we have done a lot of zebrafish models together with Wolfgang and others, and we often see severe phenotypes in zebrafish that are less pronounced in mammalians. I have to say that. So it's unknown at this point.